Okay, guys. So this is the final part of cell biology, the cell biology module on the AQA spec. We're going to be talking about these three topics. Last lesson, I finished off by talking about diffusion and what speeds up diffusion. Today, I'm going to quickly go through how to calculate surface area to volume ratio, which is something slightly different. So if we're talking about surface area to volume ratio, we're generally talking about, in biology, we're talking about heat exchange. So what it is if we have a really small organism or we have a really large organism, in relation to its surface area, which is the bit all around the outside, comparison to the volume, the stuff that makes up the entirety of the organism, the inside if you like, putting it in layman's terms, the ratio of the two is going to be lower if you are larger and higher if you are smaller. If you're smaller, you've got less volume, therefore the surface area, more of it is in contact with the environment, and therefore we get more exchange, for example with heat. And we're talking about organisms that exchange heat very well with the environment. Smaller organisms have a higher surface area to volume ratio, and therefore the exchange of heat, the exchange of whatever it is with the environment, is faster, it's more efficient. Whereas if you think about a larger organism, they have a low surface area to volume ratio, and that means that not all of their volume, not all of their mass, if you like, is in contact with the environment because the surface area doesn't cover as much of it. So if we take organisms that live in very cold environments, for example, they generally want a really low surface area to volume ratio because they don't want to lose as much temperature. So we're going to talk about how to calculate it, but this isn't every exam board, and it's certainly just with AQA, you have to be able to calculate, but it is something that you're going to have to do at A-level, so it's worth kind of knowing about. So if we look at these two shapes here, to work out the volume, we're going to do the length times by the width times by the height. And if we want to work out the surface area, what we're going to do is work out the surface area. So in a square, that's the length times the width. And then we're going to multiply that by 6 because you would have 6 sides. I'll just kind of do 1, 2, 3. But you can imagine on my diagram there, if I rotated it round, we'd have another 3 sides. So to work out the surface area to volume ratio, what we're going to do is do the surface area divided by the volume. Okay, so let's imagine that all of the lengths on this cube here are 1 centimeter. This cube, they've got to all be equal. And let's imagine here, this looks like it's about 3 times the size. So let's say these are all 3 centimeters in length. If we're working out the surface area to volume ratio, for the first one here, to work out the surface area, we've got 6 sides on the cube, and each one of them are 1 centimeter in length. So that means our surface area is 6. And if we're working out the volume of the cube, which I said here was the length times the height times the width, that's 1 times 1 times 1. 1 times 1 times 1, yes, equals 1. So then, if we're working out the ratio, then we do the surface area divided by the volume. So the surface area is 6, the volume is 1. So our surface area to volume ratio is 6 to 1, okay? Now, if it was this shape that we were working this out for, and it was larger, and we'd say it was 3 centimeters in length, that would mean that to work out the surface area, we've got 6 sides, each one is 3 centimeters in length, so 3 times by 3 would be 9, 3 by 3 is 9, so that means that our surface area is 9, and the volume is going to be just like before, length times width times height, 3 times 3 times 3, which is going to be 27. So if I know that big problem, we haven't done this, have we? So the surface area, which is 3 by 3, which is 9, and then we've got 6 sides, so 9 times 6 is 54. So if we've got a surface area of 54, we've got a volume of 27. In exactly the same way, we're going to do the surface area, which is 54, divided by the volume, which is 27. You do that, the ratio is going to be 2 to 1. So we can say our final volumes here surface area to volume ratio, sorry. Here we have a surface area to volume ratio of 6 to 1. And here we have a surface area to volume ratio of 2 to 1. So this is higher and this is lower. So it's really just a case of being able to kind of understand that because this has a higher ratio, there's going to be more exchange. But in terms of being able to calculate it, you would be expected to know how to calculate the volume, how to calculate the surface area, and to do ratios. That is sort of a basic component on most specs, not just AQA. Okay, so that's the surface area to volume ratio and why it's important. And don't forget that's slightly different to just surface area being something that increases the rate of diffusion. Now the second part of transport following the spec along here is osmosis. Now osmosis is very similar to diffusion. Please go back and watch that video if you need that recap. But we're talking about water molecules only. So osmosis is essentially the diffusion of water molecules. The tricky bit that you've got to get your head around when you're thinking about osmosis is that water molecules will, when they're in a solution where there is lots of water molecules, will be described as being the solution will be described as being dilute. And when we're talking about a solution that has very little water in it, we'll say the solution is concentrated. So that's important because if we're talking about diffusion, we often talk about the concentration difference, the concentration gradient. So if we're talking about a diffusion gradient here, concentration gradient of water molecules, this end here will be high. It will have a large concentration, a high concentration, and this side will have a low concentration of water molecules. Now, if you're just talking about the concentration of a solution rather than the particles, let's imagine we've got a glass of juice here. This has lots of solute, this has very little solute, and this has lots of water, the solvent, and this has more of the solvent. So this here is a more concentrated solution and has a higher concentration. This has a lower concentration. So that's contradictory to the concentration of the water particles. Okay? So what is best to use in your description of the movement of these particles in osmosis is dilute and concentrated. If we have a dilute solution, this solution is dilute, this solution is concentrated, that will be a lot easier to follow in your exam answers when you're describing what's going on. Water will move from where there's lots of water, a dilute solution, towards where there is little water, which is a concentrated solution, a more concentrated solution. Now, for all intents and purposes, we have to also be able to describe what happens in solutions that have more of this solute in. Now, at A level, you might go a little bit more into describing the bonding and the associations between solute and water. But at GCSE, all we really have to understand is that if you have solute in, what the solute does is it causes water molecules to stick to the solute, and therefore it's not as free to move. So if I look on the left-hand side here, here's the water molecules that I've colored in blue, and the black circles there represent the solute, like sugar or salt, something like that. Now what happens is, because we have solute molecules, these solute molecules cause water molecules to stick to them, and now they are no longer free to move. And as a result, because they're no longer free to move, we have less available water that can move across. So now if we look at these two solutions, that means that the water molecules, there is more of the water molecules on the right than there is on the left. So the water movement is from here to here. Now, initially we had the same concentration of water, but here now we've got a higher concentration in terms of free water movement here. So technically speaking, this is more dilute on the right, and this is more concentrated. So water moves towards the more concentrated solution. You might also hear about a term called water potential. Now, water potential is just the amount of water that is moving, or the ability of water to move across from one side to another. So the water potential, you'll see represented as this symbol here, is how concentrated the solution is. The lower the potential, the more likely it is that water is going to move to it. The highest a water potential can ever be is 0, and that means complete lack of solute, pure water. So if I drew my solution out on the right hand side here, only water molecules that would have a water potential of 0. That would be the highest it can be. So water will always move towards a lower water potential, one that's like more negative. Because if we start at 0 and that's the highest you can be, lower values are all going to be lower than 0, which obviously must be negative. So we can go from 0 to -1, for instance, or -2. So this water potential might be -2. This water potential 0, we get more of a movement. Now obviously it's the same for diffusion, please go back and watch that. But in terms of the rate of osmosis, where have I gone here? Let's go back down. So in terms of the rate of osmosis, if we've got more water molecules on this side, we have a higher gradient, we have a steeper gradient, so more water moves towards the more concentrated solution. Okay, there's a required practical that we do at AQA, and something very similar at Excel and OCR is an experiment where we measure the amount of osmosis that takes place in a plant cell, usually a potato. But sometimes questions ask you about kind of eggs or like other plants like carrots or whatnot, and they ask you to do a very similar experiment. So what happens in the required practical in AQA, required practical 3, is you take a bunch of potato chips that you cut using a core borer, and when you've cut them with a core borer, they all have exactly the same dimensions, so they're all exactly the same volume, because it's a kind of standardized cylinder. And then you take a ruler and you cut them all the same length, and what you do is you put them in different solutions to measure how much of a difference there has been in the length of the potato. You can use mass and you can measure the mass, it's the same principle. And what happens is, if we put them in different solutions of salt, if the concentration of the salt solution outside is more concentrated, then water will move out of the potato towards the more concentrated solution. There's still salt and sugar inside the potato, and that gives it a water potential value. It gives it a value of how readily the water will move. So if the solution is more concentrated inside the potato, water will move into the potato. But if the solution is more concentrated outside of the potato, water will move out into the solution. So therefore the potato is either going to shrink or expand depending on whether water is moving out or it's moving in. So if we put these potato chips into different solutions, all with varying concentrations, let's say this is complete water, pure water, so that might have a concentration of 0.1, 0.2, 0.3. And what I mean here by 0.3 is we're saying that that is 30% solute, 70% solvent. So it's a 0.3 molar concentration solution, if you've ever been confused about what that means and what those concentrations mean. So when we take the potato chips, we have to make sure that when we measure them at the start, we've recorded that value, and then at the end we record the difference. And then finally, we record that as a percentage difference. So the percentage difference allows us to compare how different potatoes have changed in their length. Let's imagine we start off with a potato that's 1 centimeter in length. We try to cut them all the same, but there's going to be slightly different variables. Or if we've weighed them, they're all 1 gram as a weight. If we start off with different size potatoes or different length potatoes, then what that means is we can't compare them, because it's not the same to say that a 10 centimeter potato chip has gone up by 1 centimeter and a 1 centimeter potato has gone up by 1 centimeter. That's not the same, because the 10 centimeter has, in terms of its overall size, has gone up by a lot less than the one that was only 1 centimeter to begin with. It's a much bigger change for the one that's 1 centimeter. It's doubled in size, right? So we use percentage difference in order to compare fairly different values. And what you would have done, hopefully, is plot a graph of these results. And you would have had, let's say these are positive and these are negative percentages. You would have had the concentration on the x-axis, so whatever the solution was that you plonked the potato into. And along the bottom, let's say that starts at 0, and then we've got 0.1 and 0.2 and 0.3, etc. I'm just making these values up. But we would see, hopefully, a graph that would do this. So as the concentration increased, as the concentration went up, more and more water would move out of the potato because there's a bigger gradient, there's a bigger difference in concentration. So as the water moves out, there's less and less of a percentage change, there's less of an increase. Now, if we keep increasing the concentration down here, the potato actually decreases in size. There's a negative percentage change. However, this graph also allows us to interpret something. It allows us to interpret this point here, that exact value, which on my graph there is about 4.2 in made up data world. So 4.2 moles would be the concentration where we have 0 percentage change. There's no difference. So if we've got no difference in the mass of the potato from the start to the finish, this was the potato at the beginning, and then it's the same mass at the end. That must mean the concentration outside the potato and the concentration inside the potato must be exactly the same, because there's no overall movement, there's no difference, there's no gradient. So the water movement will be overall net 0 at the concentration that's equal to the inside of the potato. So what we're doing here is we're plotting only the solutions we did. On my little kind of fantasy here, I've got 0, 0.1, 0.2, 0.3. So I plotted 0.1, 0.2, 0.3. So what this line of best fit does, it allows us to predict the value. It allows us, we didn't actually do a 4.2 molar concentration, but we can work it out by drawing this line of best fit and extrapolating it, which is something that you're sometimes asked about in terms of data and plotting it. Okay. So that's osmosis, and obviously there's a few applications where this might be important. Just remember, water absorption in animals happens in the large intestines primarily, and in plants it happens in the roots. So this is where osmosis happens. Now the final thing we're going to cover in this video, the final part of the cell biology module, is active transport. Active transport is the final mode of transport that is different to osmosis and diffusion in that it requires ATP, requires energy. So why does it require energy? It requires energy because unlike diffusion and osmosis, it isn't the movement of particles from a high concentration to a low concentration. It's the movement of particles from a low concentration to a high. So it's the opposite. So if you go back to the diffusion video I made yesterday, diffusion is the movement of particles down a concentration gradient, and we said it's passive. That means that there's enough energy in the gradient. The particles are all moving around and they're vibrating. And because they're bouncing around off each other and they're moving and vibrating, that means that there's enough energy for them to disperse and spread. But in active transport, we're trying to do the opposite. They actually want to go this way. They want to go from high to low. But what we're doing is we're putting energy into special molecules that are on the membrane, special proteins that pick up the molecules and move them from one side to the other side. So they would move from this side across to this side using the energy that activates these carrier proteins. So these carrier proteins are activated, they pick them up and they move them across, just like a crane. So that is how we overcome that. And that's only for stuff that we need to move in that is important, that we need. So in the intestines, for example, sugar is actively transported in the small intestines because we use it for respiration. So although we'd have a higher concentration inside the cells than we would in the big part in the blood than we would in the cells and the small intestine, the lumen, we still move it across because we're going to use it, we're going to need it, it's important. The other area where you'll get active transport and what you get active transport of is nitrates and minerals that are in the small intestine. So these nitrates and minerals are actively transported against a concentration gradient because they're required by the plants, and that happens in the roots of plants. So here are the roots, and in the soil we have a lower concentration of these minerals, things like potassium and magnesium that are needed in the leaves for photosynthesis and for making chlorophyll, things like that. We have a higher concentration in the roots than we do in the soil, but we still take it in because we're going to use it. It's important stuff. So that's what active transport is, and that's the difference between diffusion, active transport, osmosis. Okay, so what we're going to do now is find the questions that you could follow up with on physics and maths tutor and what I'll be looking at. So I'm going, like I am at the end of every section, to show you. Yesterday there wasn't as many questions on stem cells as there are on transport here. So I'm in the biology revision, AQA, GCSE, cell biology, and I can see these transport and cell questions at the bottom. So I might click the first section there the bit that says QP, and that'll give me some questions. I can test myself on them and then go back and use the corresponding document that says MS, which stands for mark scheme. Click that, and that'll give me the answers to the questions, so I can test myself and see if I've answered it correctly. There's a bunch of those questions, and if I was doing a different exam board, if I wasn't looking at AQA, if that's what wasn't what I was studying at school, it would be very similar questions for the different exam boards. You would just go, instead of clicking on AQA, you would go into whatever your relevant exam board was. So you go back to the biology revision, and here I've got AQA, CIE, EdXL, IGCSE, OCRE examples. So if I was to click EdXL, I know, for instance, that the transport questions are going to be in structures and functions, and then I could go into movement of substances in and out of cells, and I get very similar questions. You could even try them across exam boards if you've got lots of time and you want